Hey, welcome back. Today I want to talk to you about the physics of muscle contraction. In today's lecture, we'll talk about involuntary muscle control, isometric versus isotonic contractions, and we want to identify the motor unit. When muscles contract in response to an action potential, it produces the same results every time. As we can see here in figure 211, there's a latent phase, a contraction phase, and relaxation. During the latent phase, this is the time when excitation-contraction coupling is taking place. This topic was covered in the previous video. Contraction is where cross-bridge formation occurs and the sliding of the filaments between the actin and the myosin, allowing the sarcomere to shorten, and overall the muscle will shorten. The relaxation period is when calcium gets reabsorbed into the sarcoplasmic reticulum and the muscle tension goes back down to zero. In reality, when you engage in different activities, muscles will contract to deliver varying amounts of force in order to achieve control for that particular movement or activity. There are two types of muscle contractions. In this figure, the bodybuilder is giving an example of an isotonic contraction. He creates force against the weight, then the muscle length decreases. So overall, the muscle shortens, which changes the angle of his bones, and the weight is moved. So the force created is greater than the weight of the object. This is an isotonic contraction. Isometric contractions is when there's increasing tension on the muscle, but the muscle does not actually shorten during contraction. This is similar to what happens when you're holding plank for 1 minute. The motor unit is the somatic motor neuron and all of the muscle fibers that it connects with or innervates. The number of muscle fibers per motor unit can vary from 4 to several hundred. Muscles that control fine motor skills, like in your fingers or your eyes, have smaller motor units. Certain muscle fibers are going to be inherently stronger than others, and they can produce greater force. Large diameter fibers produce the greatest amount of force. Those would be fibers found in the legs and the glutes, so the quadriceps and gluteus maximus would be large muscle groups that create a lot of force and do the heavy lifting. So like we learned in Unit 2, there can be graded muscle responses, creating variations in the degree of muscle contraction. This is required for proper control of skeletal movements during locomotion. Responses are graded by either changing the frequency of the stimulation or changing the strength of the stimulus. That we can see here in figure 1216. The muscle's going to respond to varying amounts of stimulus. Stimulus is indicated by the red arrows along the bottom of the x-axis. When a single stimulus is applied, a single contractile response occurs. This is also known as a twitch. This is usually a procedure we conduct in the laboratory. With increasing frequency of stimuli, there is an increased tension created on the muscle, which leads to increased contractile force and eventually wave summation. This is where the muscle does not have time to completely relax, like you see here in number 2 for wave summation. The tension doesn't completely go back to zero, and as we increase the frequency of the stimulus, we can start to get what's called tetanus. This is where calcium cannot be reabsorbed fast enough by the sarcoplasmic reticulum and relaxation doesn't occur. Complete fusion or complete tetanus is the result of high-frequency stimulation. There are 3 different types of muscle in the human body. These are shown here at 100x oil. They differ in structure, location, function, and their means of activation, but all require some sort of electrical stimulation for contraction to occur. At the top is our skeletal muscle, which we talked about in the last video lecture, and we see the stacking of the fibers and the characteristic striations in the muscle. When we look at the smooth muscle example, those striations disappear. Thus, the reason why it's called smooth muscle. You don't see that striped pattern. And at the bottom, we see cardiac muscle. Again, the striations come back, but one of the things cardiac and smooth muscle have in common is that they're both involuntary muscle types. We don't have conscious control of how these muscles contract. Smooth muscle is found in the GI tract and urogenital tracts. They contract in an oblique fashion, whereas skeletal muscles tend to contract end to end, so insertion to origin, changing the angle of our bones. Cardiac muscle surrounds 4 hollow chambers that fill with blood, and smooth muscles are going to be able to stretch. They're usually surrounding hollow organs as well, but those smooth muscles will stretch to accommodate new volumes so that that volume can be held until it's moved to a different place, and you have accommodation with smooth muscle. Okay, so be able to recognize all 3 types of muscle and give structure and function details if you are given examples like this in a quiz or in a test situation. Smooth and cardiac muscle are considered involuntary due to the pacemaking ability made possible by the use of gap junctions and intercalated discs. These special structures allow for the depolarizing signal to be rapidly conducted from cell to cell so that the entire organ functions as one. These contractions are all under autonomic nervous system control. Both types have actin and myosin, but smooth muscle again lacks the sarcomeres that is so characteristic for skeletal muscle and then seen again in cardiac muscle. Smooth muscle contractions are regulated by increasing intracellular calcium concentrations through a method of calcium-induced calcium release. This is where calcium enters from the ECF into the ICF compartment, and that influx of calcium triggers the SR to release even more calcium. So that's why we call it a calcium-induced calcium release. The presence of calcium triggers cross-bridge formation through the calcium-binding protein called calmodulin. The calcium-calmodulin protein complex then binds myosin light chain kinase, and myosin cross-bridges form. Hopefully you've watched cross-bridge formation in the Pearson video assignment given to you this week. These parallel thick and thin filaments generate cross-bridge cycling in an oblique pattern. This is unique to smooth muscle. You can see here in the diagram the cells that form smooth muscle look kind of stretched and elongated, and they contract at an angle, kind of diagonally. That's what we mean by oblique. Whole sheets of smooth muscle show slow, synchronized contractions with slow, prolonged activity and low ATP energy requirements for muscle function. Smooth muscles have a unique response to stretch and allow hollow organs to fill and hold their contents. Hormones and other chemical ligands can also induce slow, sustained smooth muscle contractions. The heart is made up of cardiac muscle tissue, also an involuntary muscle, but it does have striations. These dark staining bars, which I've circled, are found only in cardiac tissue because these cells have what are called intercalated discs that contain a massive amount of protein and helps with electrical conductivity from the pacemaker. And then there's also desmosomes in here that help knit the cells together so that they can withstand the changing fluid pressures within the heart's chambers. We'll talk more about metabolism, and you should review the different types of metabolism involved with muscle contraction. This is a high energy tissue. And then we will start next. If you guys work on muscle metabolism and identify the 3 pathways used as energy sources for muscle, and then identify muscle fiber types as either fast or slow, that would complete this note set, and we can move on into cardiac, chapter 13, and that will be my next video installment for you, is the heart lecture.